Today, FDA approvals in chronic myeloid leukemia, cutaneous T-cell lymphoma, cytomegalovirus infection, and a tumor assay, and clinical holds lifted from CAR T-cell therapy trials. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved dasatinib for the treatment of pediatric patients with Philadelphia chromosome-positive chronic myeloid leukemia in chronic phase. The approval is based on results in 97 pediatric patients with CTCL enrolled across 2 studies with dasatinib. One was an open-label, non-randomized single-arm phase 2 trial, and the other was an open-label, non-randomized dose-ranging phase 1 trial. In the single-arm trial, 51 patients were newly diagnosed, and the remaining 46, 29 from the single-arm study and 17 from the dose-ranging trial, were intolerant or resistant to prior imatinib. At a median follow-up of 4.5 years, more than half of the responding patients in the treatment-naive cohort had not progressed at the time of the data cutoff. Also, the median duration of complete cytogenetic response, major cytogenetic response, and major molecular response could not be estimated. The same was true at the median follow-up time of 5.2 years for the previously treated cohort. The range of duration of response for the newly diagnosed patients was 2.5 to 66.5 months for CCYR, 1.4 to 66.5 months for MCYR, 5.4 to 72.5 months for patients who achieved MMR by month 24, and 0.03 to 72.5 months for patients who achieved MMR at any time. Among the imatinib intolerant cohort, the DOR ranges were 2.4 to 86.9 months for CCYR, 2.4 to 86.9 months for MCYR, and 2.6 to 73.6 months for MMR. In cutaneous T-cell lymphoma, the FDA approved brentuximab vedotin as a treatment for patients with cutaneous T-cell lymphoma who have received prior systemic therapy. The approval for the antibody-drug conjugate is specifically for patients with primary cutaneous anaplastic large cell lymphoma and CD30-expressing mycosis fungoides. MF and PCAL-CL are the most common subtypes of CTCL. The FDA considered data from the international open-label phase 3 ALCANZA trial, in which brentuximab vedotin induced responses lasting at least 4 months in 56.3% of patients versus 12.5% in patients receiving physician's choice of standard therapies. Results showed that at a median follow-up of 22.9 months, the median progression-free survival was 16.7 months with brentuximab vedotin versus 3.5 months with physician's choice. The objective response rate was 67% versus 20%, with complete response rates of 16% versus 2% in the brentuximab vedotin and control arms, respectively. Symptom reduction, as measured by Skindex 29, was significantly better with brentuximab vedotin versus physician's choice. The FDA has approved letermovir as a treatment to prevent cytomegalovirus infection in adult CMV seropositive patients treated with an allogeneic hematopoietic stem cell transplant. Letermovir is a non-nucleoside CMV inhibitor that targets the viral terminase complex to prevent replication. The decision was based on a significant reduction in CMV infection rates in a phase 3 study, results of which showed that 37.5% of patients treated with letermovir developed CMV by week 24 post-transplant, compared with 60.6% of those in the placebo arm. Moreover, letermovir was associated with lower all-cause mortality versus placebo at 24 weeks. The median time to engraftment and bone marrow suppression rates were similar in each group. Merck plans to make the medication available in December 2017 at a list price of $195 for a tablet version and $270 for an injection formulation. The IMPACT tumor profiling assay has been approved by the FDA. The assay, which stands for Integrated Mutation Profiling of Actionable Cancer Targets, is a tumor profiling test developed at Memorial Sloan Kettering Cancer Center. This is an in vitro diagnostic test that can identify more tumor biomarkers than any test previously reviewed by the agency. IMPACT uses next-generation sequencing to rapidly identify the presence of mutations in 468 unique genes, as well as other molecular changes in a tumor's genomic makeup. The test compares a patient's tumor tissue with a sample of normal tissue to detect genetic alterations. While it is intended to provide information on cancer biomarkers and help guide treatment options, the FDA cautions its results should not be taken as definitive when making treatment decisions. The FDA determined that IMPACT is more than 99% accurate and capable of detecting a mutation at a frequency of approximately 5%. Compared with traditional methods of detection of microsatellite instability was concordant more than 92% of the time across multiple cancer types in 175 cases. Clinical holds on 2 phase 1 trials exploring a gene-edited allogeneic CAR T-cell therapy known as UCART123 have been lifted by the FDA. Cellectis, the developer of UCART123, is working with investigators and clinical sites to obtain approval from its Institutional Review Board on the revised protocols requested by the FDA and resume patient enrollment. The agency halted UCART trials in September 2017 following the death of an elderly patient being treated for plastic plasmacytoid dendritic cell neoplasm. The FDA also cited the case of a 58-year-old patient who developed grade 3 cytokine release syndrome and grade 4 capillary leak syndrome while undergoing treatment for acute myeloid leukemia. Both conditions eventually resolved after she was treated in an intensive care unit. Cellectis is developing UCART123 as a treatment for BPDCN and AML. It was designated an investigational new drug in February 2017, making it the first allogeneic gene-edited CAR T-cell agent approved for clinical trial. UCART's process differs from the CAR T-cell therapy tisagenlecleucel in that it is allogeneic and could treat any patient using T-cells harvested from potentially any donor. Cellectis says its off-the-shelf allogeneic process is faster, easier to develop, and significantly less expensive than autologous CAR T-cell therapy. The price for a one-time treatment with tisagenlecleucel is set at $475,000. This week, we sat down with Dr. Elizabeth Mittendorf of the University of Texas MD Anderson Cancer Center to discuss new adjuvant approaches in HER2-positive breast cancer.
So I would contend that most of us believe that for your HER2-positive patients, particularly those who meet the criteria to receive pertuzumab in the neoadjuvant setting, we would prefer to have that systemic therapy administered up front. And as a surgeon, there's 2 reasons. One, it decreases the size of the breast tumor, and so can occasionally take a patient who required a mastectomy and now I can do a lumpectomy. I think a really exciting area in breast surgical oncology is that we're also seeing a similar phenomena with respect to the regional lymph node. So it takes patients who previously I would have had to have done an axillary lymph node dissection and perhaps facilitates doing sentinel lymph node evaluation alone. The other thing I think that we all as a field are beginning to embrace is the idea of assessing response to therapy to dictate the additional care, and that kind of gets to what we were discussing earlier. That pathology report after the patient receives their HER2-targeted therapy with chemotherapy in the preoperative or neoadjuvant setting, how much disease is left is going to impact the decisions for other agents in the time course over which they're given.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.